Diving into the final goal of this tabletop, which is to understand how contextual surveillance data from ongoing regional genomic surveillance can help inform outbreak interpretation. We'll do this using the ST resistance view and filtering by the outbreak strain, that ST258, and adding metadata blocks for isolation source. Here we are in Microreact back on our surveillance project. So this is named Klebsiella pneumoniae Columbia with 1,500 isolates. We are going to be using the ST resistance view here. And first up, we're going to add isolation source onto the tree as a metadata block. So click on metadata block and find isolation source. We're going to add that. And just to make it a little cleaner, I'm going to unadd everything else. All right, and we're interested in just those outbreak-associated cases. So the sequence type 258, down here in our metadata block, select ST, and we're going to filter by that 258 outbreak strain. All right, and we can see here they're really all clustering on the bottom of this tree, so they're so stacked on top of each other, you can't really get a sense of anything. So I am going to, if you move your mouse kind of further up in the phylogenetic tree, you can see when you're crossing over this intersection here, you can see this circle appear over here. We're going to right click when that circle appears, view subtree, and that's going to redraw the tree. And we could even do it again if we wanted to get rid of this, just to make it a little bit easier to see once more. That's a little tip that really helps in kind of navigating that tree that can get unwieldy when you have this many isolates. So lots and lots of different isolation source colors. What does that mean, right? So if you click on legend, you can see here the different colors and what they correspond to. With this tree zoomed into this outbreak strain ST258, we can also further apply the color column by isolation source, and that will make the dots on our phylogenetic tree correspond to the metadata block color as well. Again, and then you click on legend. So you can see there's still a wide variety of specimen sources, but the majority include blood, abdominal fluid, tracheal, and tracheal aspirates. Like we just saw, the most common isolation sources include blood, abdominal fluid, tracheal, and tracheal aspirates. Task 2 has us interpreting the phylogenetic tree to determine if sequence type 258 isolates without KPC3 are closely genetically related. For this task, we'll use the ST resistance view and filter by both ST258 and KPC3, and then look at the tree. Back in Microreact, we're going to use the ST resistance view, put them away, and we're going to filter by a couple of things. So down here in our metadata, come on over to sequence type, or ST, and again, we want that 258. And then also, we are interested in the KPC3 gene. Here we go. And we're interested in isolates without this. So we want to check 0. So these are cases of the outbreak strain ST258 without KPC3 detected. As you can see, this really cuts down on the number of cases on the tree. So one tip to kind of get a better view, because it's all smushed in there. If you hover over further to the left over here, if you hover over the line, you can see this dot appear over here, this circle. And when you see that circle, right click on your mouse, not left click, right click, and this menu will pull up. You can click view subtree, just left click on that like normal. And then it'll redraw this tree, so it's kind of more zoomed in on the nodes of interest. And you can see I sort of went one too far back, so we have this separate cluster up here that's not really of interest. So we can do it again. Right click, view subtree, and it redraws our tree for us. We can see there's only 2 ST258 isolates without the KPC3 allele detected. And these 2 isolates are genetically distinct. However, they do share a most recent common ancestor based on the location of this node here, showing how all the leaves and tips are highlighted. Like we just saw, the ST258 isolates without KPC3 detected are genetically distinct, but they are somewhat related in sharing a most recent common ancestor. For today's final task, please manipulate the project panels to identify other international isolates matching the Columbia outbreak profile. Do any isolates outside of Colombia match the outbreak profile? Look at the KP global view to identify global isolates with the same outbreak strain ST258. All right, on our KP and global view here, we're going to come down here to this timeline. You can see in this title lozenge here that our metadata panel is actually in the same kind of tab, if you will, as our timeline. So we can swap between those. So come into the metadata panel here, move it up just a touch, and we are going to filter by that outbreak strain sequence type 258. Check that. Looking at the map here, we can see two cases matching the outbreak strain ST258 are actually found outside of Colombia. So we can click on these here and control click to select more than one. And with these cases highlighted in the metadata block down here, we can see they both appear to be from the Philippines. Isolation source was blood for both of them, so they appear to be septic. And looking at the resistance genes, it appears they both have KPC2 and nothing else. So just KPC2 and ST258. As reflected here, we can see there were, in fact, two international samples, both located in the Philippines. Congratulations on reaching the end of this Microreact tabletop exercise. If you have any more questions or would like to learn more, please check out our website listed below, and thank you to the contributors that made this tabletop possible.